STS-65 was a Space Shuttle program mission of Columbia launched from Kennedy Space Center, Florida, the 8th of July 1994. The flight was commanded by Robert D. Cabana, who would go on later to lead the Kennedy Space Center crew. Backup crew mission highlights. The International Microgravity Laboratory was the second in a series of space lab flights designed to conduct research in a microgravity environment. The IML concept enabled a scientist to apply results from one mission to the next and to broaden the scope and variety of investigations between missions. Data from the IML missions contributed to the research base for the space station. As the name implies, IML-2 was an international mission. Scientists from the European Space Agency, Canada, France, Germany and Japan collaborated with NASA on the IML-2 mission to provide the worldwide science community with a variety of complementary facilities and experiments. These facilities and experiments were mounted in 2019 racks in the IML-2 module. Research on IML-2 was dedicated to microgravity and life sciences. Microgravity science covers a broad range of activities from understanding the fundamental physics involved in material behavior to using those effects to generate materials that cannot otherwise be made in the gravitational environment of the Earth. In life sciences research, a reduction of gravitation's effect allows certain characteristics of cells and organisms to be studied in isolation. These reduced gravitational effects also pose poorly understood occupational health problems for space crews, ranging from space adaptation syndrome to long-term hormonal changes. On IML-2, the microgravity science and life sciences experiments were complementary in their use of SL resources. Microgravity science tends to draw heavily on spacecraft power, while life sciences places the greatest demand on crew time. Life sciences experiments and facilities on IML-2 included Aquatic Animal Experiment Unit in Rack 3, Bio-Rack in Rack 5, Biostack in Rack 9, Extended Duration Orbiter Medical Program and Spinal Changes in Microgravity in the Center Aisle, Lower Body Negative Pressure Device, Microbial Air Sampler, Performance Assessment Workstation in the MIDIC, Slow Rotating Centrifuge Microscope in Rack 7, Real-Time Radiation Monitoring Device and the Thermoelectric Incubator both in Rack 3. Microgravity experiments and facilities on IML-2 included applied research on separation methods in Rack 6, Bubble, Drop and Particle Unit in Rack 8, Critical Point Facility in Rack 9, Electromagnetic Containerless Processing Facility in Rack 10, Free Flow Electrophoresis Unit in Rack 3, Large Isothermal Furnace in Rack 7. Quasi-steady acceleration measurement in Rack 3, Space Acceleration Measurement System in the Center Aisle, and Vibration Isolation Box Experiment System in Rack 3. Other payloads on this mission were Advanced Protein Crystallization Facility, Commercial Protein Crystal Growth, Air Force MAUI Optical Site Calibration Test, Orbital Acceleration Research Experiment, Military Application of Ship Tracks, Shuttle Amateur Radio Experiment 2. Columbia flew with an extended duration orbiter pallet and no RMS arm was installed. This was also the first flight of the payload bay door talkbox modification on Columbia and the first flight of new OI-6 main engine software. Mission overview. The second in the series of International Microgravity Laboratory payloads was launched on the Space Shuttle Columbia's STS-65 mission on the 8th of July, 1994. After remaining in orbit around the Earth for 15 days, the shuttle landed on the 23rd of July. The 7-member crew included a Japanese astronaut, who was the first Japanese woman in space. Besides NASA, the European Space Agency and the space agencies of Japan, Canada, Germany, and France sponsored experiments on the mission. Investigators from a total of 13 countries participated in research into the behavior of materials and life in microgravity. The IML-2 payload consisted of more than 80 experiments in microgravity and life sciences, including 5 life science experiments developed by American researchers. Of these, Ames Research Center sponsored 2 experiments using newts and jellyfish. Kennedy Space Center sponsored the Embryogenesis experiment, designed to study plant embryogenesis in microgravity. Life sciences research objectives. The objective of the Nude experiment was to study the early development of gravity-sensing organs. The utricle and circular are specialized organs present in the inner ears of all vertebrate animals. They contain otoliths, calcium carbonate stones, which are deposited on a gelatinous membrane that lies over the sensory hair cells. The pull that gravity exerts on the otoliths is sensed by the hair cells, and information about the gravitational stimulus is transmitted to the brain via connecting nerve fibers. The experiment was designed to determine whether otolith production and development of otolith-associated receptor cells and nerve fibers may be altered in the microgravity environment of space. The jellyfish experiment was designed to study behavior and development in space. Behavioral parameters studied included swimming, pulsing, and orientation. Study of developmental processes focused on gravity-sensing organs. The experiment also sought to determine the level of artificial gravity stimulus needed to counteract any negative effects of spaceflight. The objective of the plant embryogenesis experiment was to evaluate whether spaceflight affected the pattern and developmental progression of embryonic jellies from one well-defined stage to another. It also examined whether cell division and chromosome behavior were modified by the space environment. Life sciences payload. Organisms, adults and larvae of the Japanese red-bellied newt species were used in the newt experiment. This species was selected for study partly because the vestibular system of very young newts undergoes most of its development in a period of time equivalent to the planned mission duration. Furthermore, adult females can be induced to lay eggs by injecting them with a hormone. Their eggs develop in orbit and mature in the microgravity environment to provide scientists with a sample of embryos that have undergone early development in microgravity. Moon jellyfish served as experiment subjects for the jellyfish experiment. Both the sedentary polyp stage and the free-swimming ephora stage of the jellyfish were studied. The Embryogenesis experiment studied embryogenically competent daily cells. Hardware newt adults and larvae were housed in cassette-type water tanks in the aquarium package within the Aquatic Animal Experiment Unit, developed by NASDA, the Japanese space agency. The AAEU is a life support unit that can keep fish or other aquatic animals alive for at least 19 days in the space lab. It consists of a main unit, an aquarium package, and a fish package, each of which has an independent life support system. In IML-2, each cassette held an egg container with individual egg holes. A slow-rotating centrifuge microscope and camera system, NISEMI, developed by DLR, the German space agency, was used to examine and videotape the behavior of the jellyfish ephorian polyps at up to 15 varying levels of G and at a temperature of 28 degrees Celsius. The NISEMI provides observation of samples under variable acceleration levels between 10 to 3 and 1.5 G and a controllable temperature between 18 and 37 degrees C. Jellyfish were housed in the European Space Agency Biorack facility within Biorack Type 1 containers. For descriptions of the facility and containers, see IML-1. A refrigerator, incubator module held fixed jellyfish specimens. The RIM is a temperature-controlled holding unit flown in the shuttle middeck that maintains a cooled or heated environment. It is divided into 2 holding cavities and can contain up to 6 shelves accommodating experiment hardware. An ambient temperature recorder was placed inside the RIM. For a general description of the ATR-4, see IML-1. The PEMBSIS experiment used hardware provided by the National Space Development Agency of Japan. As part of the NASDA Life Science Cell Culture Kit, this experiment used 6 petri dish-like plant fixation chambers. The PFCs were used to hold the cultured plant cells for the PEMBSIS experiment. These containers are completely sealed. The PFCs allow plant cells exposed to spaceflight to be fixed in orbit by insertion of a chemical fixative via syringe through a septum port. Operations. Pre-flight PEMBSIS cell cultures were prepared about a week before launch. 12 chambers were filled with a semi-solid medium. 6 were transported to KSC and kept in an unlit incubator at 22 plus or minus 2 degrees Celsius until they were loaded into the shuttle. The other 6 were used as ground controls. Approximately 36 hours before launch, 148 prefertilized newt eggs were loaded into the 3 cassettes of the AAU. 4 adult newts were also loaded into the cassettes. 2 cassettes each contained 1 newt apiece, while a third contained 2. Fresh, aerated water at 24 degrees Celsius circulated continuously through the unit. A similar unit was maintained at KSC as a ground control. 24 hours before launch, 4 groups of 6 jellyfish polyps each were given iodine in artificial seawater to induce strobilization of polyps into the aphory form. Shortly before flight, the jellyfish samples were loaded into a total of 10 Nizami cuvettes containing ASW and placed in Type 1 containers. For the behavior study, a group of normal aphory and a group of aphory without statoliths were placed in the bioreactor 22 degrees Celsius incubator. The third group of aphory was placed in the bioreactor 1G centrifuge. 2 groups of polyps were used for the development study. 1 group was placed in the incubator and the other was placed in the 1G centrifuge. A similar set of equipment was maintained at the KSC ground control facility. In flight, the ambient temperature recorder is a self-contained, battery-powered instrument, approximately the size of a deck of cards. It may be placed in almost any environment to provide recording of up to 4 channels of temperature data. On flight days 6, 8, and 11, the crew carried out video observations of newt eggs to document the rate of development. The crew also made observations of the adult newts at specified times. On both the 5th and 9th days of flight, an adult newt was found dead, causing the loss of some eggs because of contamination. The remaining 2 adult newts survived the flight and were recovered live upon landing. 1 cuvette from each group of jellyfish aphory and polyps were videotaped on the rotating microscope centrifuge at intervals throughout the mission, to determine the G threshold for the swimming behavior of the aphory. On flight day 5, both the flight and ground control groups of aphory with statoliths that had been hatched on Earth were fixed. On flight day 13, 2 of the 4 groups of polyps that had been strobilation-induced were fixed. The remaining aphory and polyps were returned to Earth for post-flight analysis. To provide a comparison between flight-fixed and ground-fixed groups in the PEMBSIS experiment, the crew fixed some cultures shortly before landing. The fixative was a 3% glutaraldehyde solution. Each chamber was fixed with a 20 ml injection of fixative. Post-flight, the flight cassettes containing the newts were retrieved approximately 6 hours after landing. Some of the larvae were fixed and preserved for later analysis, while some were tested to estimate how spaceflight affected the gain of the otolith ocular reflex and measure the otolith volumes and areas of associated sensory epithelia. Living jellyfish were counted, coded, and photographed beginning at 5 hours post-flight. The pulse rate, numbers of arms, rhopalia, and statoliths were counted in each of the aphory. Those with abnormal pulsing were videotaped after landing and again approximately 24 hours later. Some of both the flight and control jellyfish were allowed to form clones, which were then examined for arm number and other structural differences. After the Embryogenesis cell culture chambers were recovered from the shuttle, specimens of living cells and somatic embryos were photographed, counted, and chemically fixed within 9 hours of landing, before their first division cycle on Earth was complete. Chromosomes were measured and compared within and among cultures. Results. Nude study. According to morphological analysis, both flight and ground controls developed at the same rates. Analysis of three-dimensional reconstructions showed that flight-reared larvae had a larger mean endolymphatic sac and duct volume and a larger average volume of otoconia within the sac when compared to similarly staged ground controls. Furthermore, the appearance of otoconia in the S was greatly accelerated in the larvae reared in microgravity. Jellyfish study of three that developed in microgravity had significantly more abnormal arm numbers as compared with 1G flight and ground controls. As compared to controls, significantly fewer three that developed in space swam when tested post-flight. Polyps budding in space produced more buds and were developmentally ahead of ground controls. Although development through budding and through metamorphosis proceeded well in space, some jellyfish are apparently more sensitive to microgravity than others, as evidenced by their abnormal arm development. Daily cell study. Cytological changes and chromosomal aberrations were seen in both flight-fixed and ground-fixed flight cells. A substantial number of binucleate cells, cells possessing two nuclei, were also found in the flight samples. The ground control samples were all uninucleate newts. At least 2 of the 4 adult newts died on the voyage. The first newt death was attributed simply to stress. The second dead newt was found by Donald A. Thomas late on Sunday, the 17th of July 1994, while checking the tanks. However, the second death was called peculiar in a comment by Dr. Michael Wiederhold, a scientist on the ground. At the time, it was said it would be difficult to remove the newt from the tank because of weightlessness, but the dead animal could contaminate the tank if left inside. The newts were Japanese red-bellied newts.